Hi, everyone. Thank you so much for having me. It's a real pleasure to be associated with a really exciting workshop just like this. So today I'm going to be talking about some work that have recently been conducting in my lab with two really talented postdocs who work with me, Eli Muller and Brennan Munn. And in general, we've been really interested in the idea of trying to understand the kinds of benefits and constraints that the thalamus, a deep diencephalic structure, imposes on the low-dimensional organization of the brain, particularly the cortex, the cerebral cortex, which we think of a lot as synonymous with the brain. And so people in this audience will be quite familiar with this idea of calculating low-dimensional gradients in corticocortical connectivity, let's say the resting state, and that this gives us a way to make contact with these really beautiful ideas. This is a figure from some prophetic work by Marcel Messelin back in the late 90s, thinking about these low-dimensional structures that are present in the cerebral cortex and how they might relate to our abilities, cognitive and otherwise. And for the fact that the cerebral cortex is so obvious visually and is quite clearly expanded over phylogeny, a lot of our models for whole brain function, things like cognition, are quite corticocentric. This is a picture of a predictive processing model where predictions are passed backwards from agranular to granular regions, and then the difference between the inputs and those predictions is propagated up the hierarchy from granular to agranular regions as prediction error. But I think it's really important that we start to think about the cortex acting in concert with the subcortical constraints imposed by regions, in particular, like the thalamus. So in this case here, I'm showing you on the left-hand side a cutaway where the thalamus in green, these diencephalic structures, you can see them sitting. There's one on the left and the right. They're sort of nestled in there with the fimbria, the hippocampus, as well as the caudate and the putamen, the basal ganglia. Right behind them are the pineal body and the superior and inferior colliculi. So that there's really, this really crucial core of gray matter structures sitting deep within the brain. And it's been known for a long time that they have different kinds of inputs to the cerebral cortex. We know that a lot of the thalamus receives input from the cerebral cortex, either from layer 6 cells or layer 5 pyramidal cells, and it projects back to them and either hits the granular layers here from a really beautiful projection from the blue brain group, or the supragranular layers. Which is where a lot of the feedback connections from agranular regions are actually received by the cortical inputs. And so traditionally, there are lots of different ways to think about the thalamus. Murray Sherman's popularized the idea of first order, i.e. regions that project, let's say, a sensory signal straight forward into the brain, into the cortex, and higher order, which are regions of the thalamus that, instead of receiving sensory input, receive input from the cortex and then feed back to the cortex. You could think of them as a sort of embedded loop within the central nervous system. The dichotomy that I'm quite fond of, and I'm going to speak a lot more about today, is an idea popularized by Ted Jones, which is the idea of a core matrix architecture. So in this case, the core nuclei are sets of individual cells that are present within pretty much every single larger cellular group or nuclei within the thalamus. And they typically receive inputs from either the sensory apparatus or the spinal cord or the cerebellum, and they project forward into the granular layers of the cortex in a quite targeted way. They're interdigitated within each of these different nuclei in a mixture are actually a different population, which Ted Jones called the matrix population. And these instead project up to the supragranular regions of the cortex, and they project in a quite diffuse manner that often crosses obvious functional boundaries. Okay, and they're going to project up and hit the supragranular layers and hit different kinds of cells and different regions within those cells in the cortex, and can theoretically push them into a different processing mode. So one of the things that we're really interested in is trying to work out what having a system that has these different levels, these types of organizational principles in it, could buy you at the systems level. Now, fortunately, there's a way in here, which is that these different thalamic populations express different calcium binding proteins. So the matrix cells express calbindin and to a lesser extent calretinin, and the core cells, at least in non-human primates and humans, express parvalbumin. And so what we can do is go to the Al-len Brain Atlas, which is a repository of 6 individuals that post-mortem had their brains donated to science, and then every single region of their brain was essentially conducted, a genetic test was conducted that looked at the levels of mRNA expression of a range of different genes. And what we can essentially do then is look at thalamic regions of interest, and we can go in and we can ask what the relative expression of calbindin in blue or parvalbumin in green was. And what that lets us do is essentially create an estimate of the likely population level activity within those 2 different classes. And you can see that across the thalamus, there's a sort of general gradient of expression of these two different calcium binding proteins, which we can use. And so when we then correlate the time series with the time series of a set of 400 regions from the cerebral cortex, in this case, it's a 7 Tesla data set, 60 subjects, which we was provided by one of our collaborators, Luca Kochi from Queensland. We're actually able to show that the parvalbumin cells or voxels within the thalamus were much more likely to have strong correlations with visual, temporal, and somatomotor areas, whereas the calbindin population were much more likely to correlate with posterior cingulate, regions of the frontal, medial frontal cortex, as well as the lateral frontal and lateral parietal cortex. And then down the bottom, I'm just showing the relative mean within each of the different 17 subnetworks of the Yo17 parcellation. And so this little map at the top should actually look quite familiar to people in this audience. Namely, we have a Margulies alert. We found through completely different set methodologies, we can take genetic architecture of the thalamic voxels, and we can then look at their time series, and we can actually recapitulate this primary resting state gradient, which we would normally find by, let's say, getting vertex to vertex connectivity measures and then performing some kind of dimensionality reduction on that data. It's worth mentioning as well here that this result survives correction for spatial autocorrelation, in which we use the spin test to populate a null distribution. And so, so we thought that was really interesting, that we could basically take thalamic architecture and then recapitulate cortical connectivity patterns in a low fashion. Brandon Munn, one of the really talented postdocs in my lab, was really interested in whether or not this pattern of sensory to associative connectivity that was recapitulated in the thalamus might also relate to the kinds of timescales that the system is able to process. And the idea here is that sensory regions are able to sort of refresh really quickly, whereas the regions like associative regions, frontal parietal cortex, are actually able to process information over longer timescales. And so to test this hypothesis, he calculated the Hurst exponent on each cortical parcel, and then related that to the relative connectivity between the matrix thalamus, i.e. the calbindin positive thalamic cells, and the parvalbumin positive, or the core thalamus. And we essentially found a positive correlation that again survived the spin test. Eli Muller, another really talented postdoc in my lab, was really interested in making contact with this notion of the attractor landscape. We find this a really intuitive and rich framework in which to view the idea of state-space dynamics. The idea is that you can envisage the particular brain state at a point in time as a ball sitting upon a topography and attractor landscape, where the depth of the well that the ball is currently sitting in tells you something about the stability of the current state, whereas the flatness tells you about the ability for that ball to roll to a new state. And so the idea might be that if you had the core system driving really hard, it might be something like the depth of this well would decrease, so you stay stuck there, right? Whereas if you wanted to get to a new state, what would actually be really beneficial is if you could squirt in a little bit of variability, if you could increase the temperature of the activity patterns that were allowing you to reach that new attractor, and that might be this green ball here, which can then overcome that activity, that energy, and then find its way into this new state. And Eli was essentially able to find evidence for this phenomena. The idea here is that he calculated time varying functional connectivity with a range of different windows. But for each window length, he essentially calculated the variability of the dynamic connectivity pattern across all of the windows for each region. And then we were able to show that the regions that had the most variable dynamic connectivity are the ones that were able to configure into multiple different zones, multiple different types of configurations, were the ones that were supported by the matrix more than the core thalamus, which again, I think fits with the intuition that the matrix thalamus was able to buy you this ability to reach a new brain state that you couldn't have otherwise reached. In the next little part of the talk, as a sort of a way to kind of conclude and wrap up some of these ideas, I wanted to talk to you about some of the computational work that Eli and Brandon have been conducting in the lab that's essentially trying to put some mechanistic basis underneath a lot of these ideas that we've been discussing so far. The idea is what we can do is we can take a really well-established corticothalamic neural mass model, which really does look an awful lot like the core thalamic connectivity pattern, where you have an excitatory cortical population projecting down to a relay nucleus in the thalamus and then projects back up to the population in the cortex in a kind of little feed-forward loop. And then what Eli was able to do is then put in a variable amount of diffuse connectivity, which would represent something like the matrix thalamic input. And as he turned it up and down, the idea is he could start to work out what was the benefit of the system being organized in that fashion. You could think about this as analogous to heating up a glass of water. And the idea is that as you slowly heat up the glass of water, more and more regions that fall on the right tail of the distribution of temperatures within that glass will actually then be able to think that the temperature is hotter than it actually is. And then they can essentially move into a different kind of a brain state, a different kind of a state in the case of water, but that might be a different brain state in the model that we're conducting. So you can think about this as the system being enslaved to a lower attractor, but if we squirt in enough diffuse coupling, now all of a sudden a small group of regions can kind of break away from that enslavement and do something a little bit different, let's say fire at a higher rate, or congregate into a small neural assembly that might allow you to process some information in a contextual fashion. So we think that this might be the really key computational benefit of having a system that has this ability to project in a diffuse manner and really sort of raise the temperature of the cortical population without necessarily passing a message between regions. And so this slide is just to demonstrate that you can take this parameter, sweep it through a range, and take the system from one in which there's a very little activity and everything's quite subcritical and damped, to a region of saturation where everything's sort of spiking away, but in a really information poor way. And the transition between these two, which we call the quasi-critical regime, is really crucial because a little bit more diffuse coupling allows you to actually reconfigure the brain state in this model and process information in completely unique ways. One example, there's plenty more in the paper if you're interested, but one example that I think is really telling is that if you take these regions, the different nodes in each of these different regions, and you ping them with a identical input in each case, what you can find is that in the subcritical zone, regions essentially all come back to their baseline at the same rate, whereas in the quasi-critical zone, you actually start to see a separation where the regions that were further to the right in our original model, the closer to that bifurcation, the ones that had sort of realized a higher temperature, were actually able to move into a completely different functional mode. And you get this really interesting separation of timescales that can allow you to have sort of subtle interactions amongst regions that you couldn't have in the subcritical or the saturated zone. I don't have time to get into it here, but you actually see a lot of the same signatures of complex adaptive dynamics that people have found in different systems level neuroscience experiments. So for example, the network becomes more integrated and more low dimensional as you move into this quasi-critical regime, which I think is really important. And it actually recapitulates some work I did with Luca Cocci and colleagues, where we took a Latin squares task, which is a little bit like Sudoku, and we're able to show that the low dimensional state space dynamics in the cortex are actually quite similar to a lot of the dynamics that were happening in the thalamus. In this case, we were partitioning the thalamus into different nuclear compartments rather than genetic compartments, but I think the same intuition is consistent in these two different approaches. And so essentially what we could do, right, is we could take now data from, in this case, an end-back task and rest, and we could ask where in that that landscape of diffuse, relatively low diffuse to relatively high diffuse coupling does our brain fit? And essentially, Eli had to create his own model fitting algorithm, but you can see that we find evidence that the end-back task is better explained by a higher level of diffuse coupling and a more quasi-critical brain, whereas the resting state was more consistent with a subcritical or maybe lower side of quasi-critical. So in summary, I really want people, at a high level to be thinking the cortical structures that we see, the cortical organizing principles that we see, ought to have relationships with these really highly conserved subcortical systems, in particular the thalamus, but there's a range of others, the striatum, the hippocampus, the amygdala, the superior colliculus, the hypothalamus, that are really worth thinking about when we start thinking about these sort of higher level systems and how they're coordinated. And with that, I'd like to say thank you, and please, if you're interested, check out the work on bioRxiv. A huge thanks to Eli Muller and Brendan Munn, who did a lion's share of the work here. Thank you very much.